press the bell icon on the YouTube app and never miss another update.
Hi everyone, I welcome you all for this session. In this session, we are going to understand the concepts of biotechnology, the first chapter under the unit biotechnology, the biotechnology principles and processes. So before jumping into the concepts exactly, let us understand the few terminologies which are actually concerned to this particular topic. So we know that there are the familiar terminologies such as science, engineering and technology. Science, engineering and technology. So what exactly is the meaning of science, engineering and technology? We need to understand this particular terminologies. So wherein the science which refers to the basic concepts, basics, principles and facts of nature. So understanding or studying the basic principles or the facts of nature, which is referred as science. So wherein engineering, so know that the literal meaning of engineering is to manipulate or modify. So whatever the basic principles and facts which are existing in the nature, and understanding all these principles and the processes or the facts, and if you are able to manipulate it or modify it for further applications or any other the processes, then particular the modification will be referred as engineering. And then what exactly is technology? So after modifying some basic principles or the facts or the processes which are existing in the nature, and if you are applying it for the benefit of mankind, then it will be referred as technology. So technology refers to applications. So today we are into various technologies. The technologies has been into various fields, such as it may be the aerospace technology, or it may be electrical or electronic technologies, or it may be some the biological technologies also. So wherein the biotechnology, it refers to understanding the basic principles, facts which are actually confined to the living entity, and modifying them or manipulating them for the welfare of the mankind. So all those, the use of living organisms, it may be prokaryotic or the microorganisms, what we say, or even it may be the eukaryotic organisms, and there may be certain biomolecules also. So use of all those biomolecules, modifying them and having an applications of these living organisms for the benefit of mankind will be referred as biotechnology. This biotechnology, the term is not the recent one. It's not the very the modern terminology. So wherein the biotechnology, which actually uh, somewhere around it goes back around some 300 BCs also. So wherein the use of simple microorganism for simple processes, such as it may be the formation of curd, or it may be the production of the wine, such processes were also referred as the biotechnological processes. So wherein there are different eras of this particular biotechnology, let us see what are those different stages of evolution of this particular technology. See, it can be into three different eras of biotechnology, such as the traditional biotechnology, or it may be conventional biotechnology, and it may be modern biotechnology, what we are going to discuss in this session. Right. So, different eras of biotechnology. So, it starts with traditional biotechnology, then the conventional biotechnology, then the modern biotechnology. So, what is this traditional biotechnology? So, traditional biotechnology, which refers to all those simple processes, simple microbial processes. So I just mentioned like simple microbial processes like conversion of the milk sugars, the lactose into lactic acid, in the sense the production of the curd, production of the wine. So all these process, simple microbial processes, they will be referred as traditional biotechnological processes. And what exactly is conventional biotechnology? Conventional biotechnology, this comprises certain techniques or the methods. So wherein there will be improvement of crops, and the technique will be referred as breeding techniques, breeding technology, otherwise called hybridization technology. So understanding the characteristic of different plants. So wherein we can combine the characteristics. We can have an hybrid character by combining certain the elite donors. So, for example, a plant may be having high protein content, ability to produce high protein content. The other plant may be having their disease resistance. And these two characters, if at all we need to combine into a single plant, then we have a particular technology which is it's possible that the protein producing ability as well as the disease resistance ability can be combined into single plant. So that's what which is referred as breeding technology or the hybridization. So which is into conventional biotechnology. Then what exactly is modern biotechnology? So modern biotechnology, which actually refers to recombinant DNA technology. So recombinant DNA technology, otherwise it is also called as genetic engineering, and it can also be referred as transgenic technology. So we know that there are the two main types of reproduction, that is asexual reproduction as well as sexual reproduction. So, wherein asexual reproduction, which preserves or conserves the genetic content or the characteristics of the genetic material. So, wherein the sexual reproduction enables to produce variations. It helps in formation of different characteristics or combination of characteristics. See, conventional biotechnology, which enables us to bring two characteristics from two different plants into a single plant. And breeding is possible only between the related species. Only between the related species. And wherein breeding is also having certain limitation that in case of breeding, there is all possibility that even the undesirable characters may also enter into the progeny or the hybrid plant. So in such cases, so wherein the undesirable characters cannot be prevented or it cannot be avoided. So, breeding is a technique wherein we can bring the two allied characters, but there is all limitation that So even the undesirable properties of some plants can also enter into the progeny. Whereas the modern biotechnology, which refers to recombinant DNA technology or genetic engineering or transgenic technology. So this is an greatest advancement in the biological sciences. So wherein there is a possibility of transferring a desired gene, a single gene from any cell to any cell. Recombinant DNA technology, which enables us to transfer a single desired gene from any cell to any cell. I mean to say, we can transfer a gene from a prokaryote to eukaryote in general. So it may be bacteria to plant, plant to bacteria, or plant to fungal cell, or plant bacteria to fungal cell, or from bacteria to animal cell, or animal to plant cell, or animal to even the bacterial cell. So across the domains, there is a possibility of transferring gene. So this gives us an opportunity, so wherein we can overcome the limitations of this conventional biotechnology, what we call it as the breeding technology or the hybridization. So in hybridization, so there was no possibilities and there was a limitation that we are unable to, we are inevitable, so wherein we cannot avoid the transfer of the undesirable genes. So whereas recombinant DNA technology, so which enables us to find out a novel gene, and that particular single gene, a novel gene, can be isolated and it can be transferred into the target organism. So for example, this is the donor cell or the bacteria. From this donor cell, so we can transfer a desired gene into the host cell. So our desired gene can be transferred into the host cell. So there are two advantages. So the first advantage is, so we are very particular that whatever the gene, desired gene is required, only that particular gene can be transferred into the host cell. And the second, or the most important advantage is, so we can transfer gene across the domains I told. That means from prokaryote to eukaryote or eukaryote to prokaryote, across any kingdom or any organisms. So there is a possibility of transfer of genes. So this is what most important advantage of the recombinant DNA technology. Then why the technology is referred as recombinant DNA technology? So here, if you are possibly transferring a gene from bacteria, so you may be very familiar with example, that is the BT cotton or the transgenic Bacillus thuringiensis cotton. So which is a insect resistant plant. So wherein the gene from Bacillus thuringiensis was transferred into the cotton plant, making that cotton plant naturally resistant to insects. So the plant is basically naturally, it's not having a gene called crystal protein gene. But whereas the crystal protein gene, which is actually present in the bacteria, is isolated and it is transferred into the plant. So now the plant is having a new gene which is not actually native one. So as the plant is having a new combination of DNA, hence the technology is referred as recombinant DNA technology. Then it is also referred as genetic engineering. So I just mentioned in the introduction that the engineering, the term literally it refers to manipulation. So it may be manipulation of anything. For example, we have different branches of engineering, such as it may be computer science engineering, mechanical engineering, electrical, electronics, civil engineering, etc. So even we have the genetic engineering. So you may not be aware or may be aware of, even there is a branch of the course, engineering course, so which is referred as genetic engineering, which refers to manipulation of genetic material, which refers to modification of the genetic material for further applications, for further improvement of certain characteristics of living organisms. So that the overall the technology which enables us to modify, manipulate the genetic material is referred as genetic engineering. It's called transgenic technology. Transgenic. So trans refers to it is a transfer of gene or a gene from other source. So wherein we are able to transfer a gene from one cell to another cell, from bacteria to plant, from plant to bacteria. So as there is a possibility of transferring gene or transportation of gene from one cell to another cell, so the technology is referred as transgenic technology. Right. So there are different definitions which can be entitled for defining the biotechnology. So whereas European Federation of Biotechnology, EFB, so European Federation of Biotechnology has given a proper definition for biotechnology. So where it goes like this. So it is integration of natural science, organism, cells, parts thereof, and molecular analogs for products and services. So, according to European Federation of Biotechnology, the statement or the definition for biotechnology goes like this: it is the integration of natural science with the organisms or the cells or parts thereof. The cellular parts are the molecular analogues for products and services for the betterment of the mankind is referred as biotechnology. So it is the integration of science and technology in general. So wherein uh, the living entities, it may be microorganisms or eukaryotic cells or their part cells or the molecular analogues are utilized for the benefit such as the products or the services to the mankind. Right. So there are various core technologies or the methods of RDNA technology. So among all techniques or the technologies, there are two core techniques of biotechnology, principles of biotechnology. The two main core techniques are genetic engineering, and the second is maintenance of sterile ambience. See, the two main techniques. So of course, there are other the techniques also. Among all those techniques, the two core techniques which enables the RDNA technology are the genetic engineering and the maintenance of sterile ambience. So I just told you, like the RDNA technology can also be referred as genetic engineering. So which makes the core the step or the technique of the modern biotechnology. So genetic engineering refers to the chemical modification of the genetic material. So it is the modification or manipulation of genetic material. So generally, the DNA is the genetic material. In some cases, it may be RNA. So wherein we are going to modify this genetic content and we are going to introduce that particular character into the desired organism or the host organism. And maintenance of sterile ambience. Sterile ambience refers to micro-free environment or contamination-free. Micro-free or contamination-free environment. So most of the applications of RDNA technology are into industrial productions. So whereas the production of such products such as the vaccines or the antibiotics or the enzymes or some the immunological products such as interference, etc. So we are going to produce all these biomolecules in a larger scale. So we are going to extract those particular the biomolecules in a industrial scale. So during that, a micro-free or a contamination-free environment is very much essential in order to obtain the desired product in a larger scale. So making an environment micro-free, so wherein we can grow only the desired type of organism. It may be the desired microorganism or it may be desired eukaryotic cell. So wherein the undesired organisms has to be prevented by growing in the industrial, the production or the whereas the medium what we are taking for the production of the desired product in a larger scale. So maintenance of sterile ambience becomes most important the method in RDNA technology. Right. The first genetic engineering experiment. First RDNA recombinant DNA experiment or first genetic engineering experiment. The first genetic engineering experiments were conducted by scientists by name Herbert Boyer and Stanley Cohen. So Herbert Boyer and Stanley Cohen, so these two scientists carried out the first genetic engineering experiment. So wherein they isolated an antibiotic resistant gene from a bacterium called Salmonella typhi. So antibiotic resistant gene from Salmonella typhi was ligated to native plasmid. So native plasmid in the sense the plasmid of Salmonella typhi itself. So plasmid, you know that it is a extra chromosomal, circular, double-stranded, autonomously replicating bacterial DNA. So the antibiotic resistant gene from Salmonella typhi was ligated to the plasmid of the same Salmonella typhi, ligated to native plasmid and transfer to the related microorganism, that is Escherichia coli, E. coli bacterium. So they were able to successfully transfer an antibiotic resistant gene from Salmonella typhi into the Escherichia coli. So this was the first genetic engineering experiment which was conducted by Herbert Boyer and Stanley Cohen in the year 1972. Right. So there are so many tools which are actually important in transferring a gene from one cell to another cell. So it's very tedious process wherein you can just imagine a cell is microscopic, right? So we cannot visualize the cell through our naked eyes. So we need to take the help of microscope. So wherein we can observe certain structures, not in detail, of course. So in detail, again, we need to go the higher the type of microscope, such as the electron microscope. So wherein we can study the internal structures of the cell. Cell is microscopic, and inside the cell, in case of eukaryote, there is a nucleus, and inside the nucleus there is a DNA, and from DNA, out of so many types of the genes, so we need to find out which is our desired gene. We need to isolate that desired gene, and we need to transfer the desired gene into the host cell. So we can just imagine what would be the difficulties and uh, the tedious processes which are actually involved in this technology. It's not very just a simple statement that it is a transfer of gene from one cell to another cell. ಅಷ್ಟು ಸಿಂಪಲ್ ಆಗಿರೋದಿಲ್ಲ. ತುಂಬಾ ಟೆಕ್ನಾಲಜೀಸ್ ಬೇರೆ ಬೇರೆ ಸ್ಟೆಪ್ಸ್ ಇರುತ್ತೆ, ಬೇರೆ ಬೇರೆ ಮೆಥಡ್ಸ್ ಇರುತ್ತೆ. ಸೊ ಎಲ್ಲಾ ಮೆಥಡ್ಸ್ ಏನೇನಿದೆ, ಏನೇನ್ ಟೂಲ್ಸ್ ಬೇಕಾಗುತ್ತೆ ಅನ್ನೋದನ್ನ ನಾವು ನೆಕ್ಸ್ಟ್ ಪಾರ್ಟ್ ಅಲ್ಲಿ ಡಿಸ್ಕಸ್ ಮಾಡ್ತಾ ಇದೀವಿ. Right. Tools for RDNA technology. Right. So tools for RDNA technology. I'll just make it very simple by giving you an example, a familiar example. So it's a technology, mobile technology. You are all very familiar and expert with what are all the new tools and the new apps which are actually used. Simple Android mobiles ಎಲ್ಲರೂ ಯೂಸ್ ಮಾಡ್ತಾ ಇರ್ತೀರಾ. ಸೊ ಅರ್ಲಿಯರ್ ನಮಗೆ ಬೇಸಿಕ್ ಟೈಪ್ ಆಫ್ ಮೊಬೈಲ್ ಫೋನ್ಸ್ ಇತ್ತು. ಅದಕ್ಕಿಂತ ಮುಂಚೆ ನಾವು ತಗೊಂಡ್ರೆ ಆಕ್ಚುಲಿ ಲ್ಯಾಂಡ್ಲೈನ್ ಫೋನ್ಸ್ ಅಂತ ಇತ್ತು. ಸೊ ದೆರ್ ಇಸ್ ಎನ್ ಎವಲ್ಯೂಷನ್ ಇನ್ ದಿಸ್ ಪರ್ಟಿಕ್ಯುಲರ್ ಟೆಲಿಫೋನಿಕ್ ಟೆಕ್ನಾಲಜಿ. ಸೊ ಯಾವ ತರ ಮೊಬೈಲ್ ಬೇಸಿಕ್ ಸೆಟ್ ಇಂದ 3310 ನೋಕಿಯಾ ಅಂತ ಇತ್ತು ಆಕ್ಚುಲಿ ಸ್ಟಾರ್ಟಿಂಗ್ ಅಲ್ಲಿ ಮೋಸ್ಟ್ ಪಾಪ್ಯುಲರ್ ಹ್ಯಾಂಡ್ ಸೆಟ್. ಅದಾದ್ಮೇಲೆ ಏನೇನ್ ಬಂತು, ಯಾವ ತರ ಕಲರ್ ಸೆಟ್ ಬಂತು, ಅದಾದ್ಮೇಲೆ ಏನೋ ಟಚ್ ಸ್ಕ್ರೀನ್ ಮೊಬೈಲ್ಸ್ ಬಂತು. ಇವತ್ತು ಆಂಡ್ರಾಯ್ಡ್ ಮೊಬೈಲ್ಸ್ ಅಂತ ಹೇಳ್ತೀವಿ, ಐಫೋನ್ಸ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಸೊ ದರ್ ಇಸ್ ಅನ್ ಎವಲ್ಯೂಷನ್ ಆಫ್ ದಿಸ್ ಟೆಕ್ನಾಲಜಿ. ಸೊ ಇಲ್ಲಿ ಒಂದು ಹೈ ಎಂಡ್ ಮೊಬೈಲ್ ಅಲ್ಲಿ ಏನೆಲ್ಲ ಫೀಚರ್ಸ್ ಇರುತ್ತೆ, ಅಡ್ವಾನ್ಸ್ ಮೊಬೈಲ್ ಅಂತ ಹೇಳ್ಬೋದು. ಸೊ ಎರಡು ಮೊಬೈಲ್ ಅಲ್ಲಿ ಒಬ್ರ ಹತ್ರ ಒಂದು ಪರ್ಟಿಕ್ಯುಲರ್ ಮೂವಿ ಇದೆ ಅನ್ಕೊಳ್ಳೋಣ, ರೀಸೆಂಟ್ ಮೂವಿ ಯಾವ್ದು, ಇನ್ನೊಬ್ರಲ್ಲಿ ಇಲ್ಲ. ಏನ್ ಮಾಡ್ತೀರಾ? ಐ ನೀಡ್ ನಾಟ್ ಎಕ್ಸ್ಪ್ಲೇನ್, ಇಮಿಡಿಯೇಟ್ಲಿ ಏನ್ ಮಾಡ್ಬೇಕು ಅನ್ನೋ ಟೆಕ್ನಾಲಜಿ ಆಪ್ ಗೊತ್ತಿದೆ. ಸೊ ಎರಡು ಮೊಬೈಲ್ ನ ಕನೆಕ್ಟ್ ಮಾಡ್ತೀರಾ ಟು ಪರ್ಟಿಕ್ಯುಲರ್ ಆಪ್, ಏನದು ಶೇರ್ ಇಟ್ ಅಂತ ಹೇಳ್ತೀವಿ, ಅಥವಾ ಇನ್ನು ಅಡ್ವಾನ್ಸ್ ಏನು ಬೇರೆ ಬೇರೆ ಆಪ್ಸ್ ಬರ್ತಾ ಇದೆ. ಸೊ ಎರಡು ಮೊಬೈಲ್ ಕನೆಕ್ಟ್ ಆಗುತ್ತೆ. ಸೊ ಈ ಮೊಬೈಲ್ ಅಲ್ಲಿ ನೀವು ಫೋಲ್ಡರ್ ಓಪನ್ ಮಾಡ್ತೀರಾ, ಆ ಫೋಲ್ಡರ್ ಅಲ್ಲಿ ಸುಮಾರು ಫೈಲ್ಸ್ ಇರುತ್ತೆ. ಆ ಫೈಲ್ಸ್ ಅಲ್ಲಿ ಯಾವ ಫೈಲ್ ಬೇಕಂತ ಕ್ಲಿಕ್ ಮಾಡ್ತೀರಾ. ಕ್ಲಿಕ್ ಆದ್ಮೇಲೆ ಅದನ್ನ ಸೆಂಡ್ ಅಂತೀರಾ, ಇಲ್ಲಿ ರಿಸೀವ್ ಕೊಡ್ತೀರಾ, ಈ ಫೈಲ್ ಬಂದ್ಬಿಟ್ಟು ಇಲ್ಲಿ ಟ್ರಾನ್ಸ್ಫರ್ ಆಗುತ್ತೆ. ಸೊ ಔಟ್ ಆಫ್ ಸಮ್ 100 ಫೈಲ್ಸ್ ಯು ಆರ್ ಏಬಲ್ ಟು ಟ್ರಾನ್ಸ್ಫರ್ ಓನ್ಲಿ ದಿ ಡಿಸೈರ್ಡ್ ಫೈಲ್ ಫ್ರಮ್ ದಿಸ್ ಮೊಬೈಲ್ ಟು ದಿಸ್ ಮೊಬೈಲ್. ಸೊ ಇದು ಒಂದು ಟೆಕ್ನಾಲಜಿ ಇದೆ. ಸೊ ಅದೇ ತರ ಇಲ್ಲಿ ಬಯೋಟೆಕ್ನಾಲಜಿಯಲ್ಲಿ ಅಥವಾ RDNA ಟೆಕ್ನಾಲಜಿಯಲ್ಲಿ ಒಂದು ಪರ್ಟಿಕ್ಯುಲರ್ ಆರ್ಗ್ಯಾನಿಸಮ್ ಇಂದ, ಅಂದ್ರೆ ನಾವು ಡೋನರ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಸೊ ಡೋನರ್ ಇಂದ ಇನ್ನೊಂದು ಆರ್ಗ್ಯಾನಿಸಮ್ ಏನ್ ಹೇಳ್ತೀವಿ, ರಿಸಿಪಿಯಂಟ್ ಆರ್ ದಿ ಹೋಸ್ಟ್ ಸೆಲ್ಗೆ ಆ ಡಿಸೈರ್ಡ್ ಜೀನ್ ನ, ಪರ್ಟಿಕ್ಯುಲರ್ ಜೀನ್ ನ ನಾವು ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡ್ತಕ್ಕಂಥ ಪಾಸಿಬಿಲಿಟಿ ಇದೆ. ಸೊ ಇಲ್ಲಿ ಮೊಬೈಲ್ ಇಂದ ನೀವು ಒಂದು ಮೊಬೈಲ್ ಇಂದ ಇನ್ನೊಂದು ಮೊಬೈಲ್ಗೆ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡೋದಕ್ಕೆ, ಒಂದು ಫೈಲ್ ನ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡೋದಕ್ಕೆ ಅಲ್ಲಿ ಕೆಲವೊಂದು ಟೂಲ್ಸ್ ಬೇಕಾಯ್ತು, ಆಪ್ಸ್ ಬೇಕಾಯ್ತು. ಸೊ ಪೇರಿಂಗ್ ಮಾಡ್ಬೇಕಾಯ್ತು, ಒಂದು ಪ್ರಾಸೆಸ್ ಇದೆ, ಟೆಕ್ನಿಕ್ ಇದೆ, ಒಂದು ಪ್ರೊಸೀಜರ್ ಇದೆ ಫೈಲ್ ನ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡಬೇಕು ಅಂತ. ಸೊ ನಾವು ಅನ್ಕೊಂಡ ತಕ್ಷಣ ಫೈಲ್ ಟ್ರಾನ್ಸ್ಫರ್ ಆಗ್ಬೇಕಂತ ತಕ್ಷಣ ಆಗೋದಿಲ್ಲ. ಸೊ ಎರಡನ್ನು ಪೇರ್ ಮಾಡ್ಬೇಕಾಗ್ತಾ ಇದೆ. ಇಲ್ಲಿ ಓಪನ್ ಮಾಡಿದೀವಿ, ಲಿಸ್ಟ್ ಆಫ್ ಫೈಲ್ಸ್ ಬರ್ತಾ ಇದೆ, ಆ ಫೈಲ್ ನ ಕ್ಲಿಕ್ ಮಾಡ್ತಾ ಇದೀವಿ, ಡಿಸೈರ್ಡ್ ಫೈಲ್ ಯಾವ್ದು ಬೇಕು ಅಂತ ಸೆಲೆಕ್ಟ್ ಮಾಡ್ತಾ ಇದೀವಿ, ಅದಾದ್ಮೇಲೆ ಅದನ್ನ ಸೆಂಡ್ ಮಾಡ್ತಾ ಇದೀವಿ, ರಿಸೀವ್ ಮಾಡ್ತಾ ಇದೀವಿ. ಸೊ ಇಷ್ಟೆಲ್ಲ ಪ್ರಾಸೆಸ್ ಇದೆ ಇಲ್ಲಿ ಕೂಡ. ಸೊ ಸಿಮಿಲರ್ಲಿ, so if at all we need to transfer a desired gene from donor cell to the recipient cell or the host cell, so there must be certain processes, there must be certain techniques, there must be certain tools for this technology. So in order to transfer the desired gene, single gene, first we need to isolate the DNA, then we need to find out, we need to cut that particular specific required segment of the DNA, and then we need to transfer. For transferring, we need to we need to have certain the mechanism or certain tools for transferring. And after that, if the whether the desired gene is transferred into the host cell or not. So all these methods, what we make, what it comprises of, the tools, all these requirements, right? So basically, RDNA technology, wherein we are actually isolating the DNA, cutting specific segment of DNA, then we are ligating it into some particular structure, what we call it as vector, and then we are transferring it into another host cell. So wherein we are modifying, we are bringing new characteristics into the host cell. So in general, in a common terminology, terminology, the RDNA technology is also referred as molecular tailoring. Molecular tailoring. So tailor require scissor, he require the needle for stitching, and all these are the tools where basic tailor require. Similarly, in order to have a recombinant DNA, so we need to have certain tools. So let us see what are those tools for RDNA technology. So basically, the most important tools are the enzymes. So in these enzymes, again, we have most important enzymes such as restriction enzymes, then the ligases, polymerase enzymes, and the other group of enzymes can be called as DNA modifying enzymes. So that's about the first important requirement, and the second most important tools are referred as vectors. Third, the host cell. Fourth, the desired gene, and the bioreactors. So the important tools which enables the technology, or which are mandatory for RDNA technology, are the enzymes, then the vectors, then the host cell, the desired gene, and the bioreactor. Let us understand one by one, starting with enzymes. So enzymes, basically, these are the proteins which are involved in the biological reactions. They are referred as biocatalysts. So among enzymes, the most important enzymes which enable the RDNA technology are the restriction enzymes. What exactly these are the enzymes, restriction enzymes? See, the restriction enzymes basically these are the class of the enzymes called nucleases. Nucleases are the enzymes which act upon either DNA or RNA. If the enzymes act on DNA, they are referred as DNAases. And if the nucleases are acting on the RNA, they are referred as RNases. DNases are the nucleases which act on DNA, and RNases are the nucleases which act on RNA. And further, the DNases may be general DNases and restriction enzymes. So the general DNases such as S1 nuclease, so these are the group of enzymes. So wherein the enzymes can act on single-stranded DNA or double-stranded DNA at any part, in the sense any point of DNA. Whereas the restriction enzymes, so there is a specific name restriction. These are the DNases which act on the DNA at specific sites. The DNases which act on double-stranded DNA particularly at specific sites, and those specific sites are called recognition sequences. So very simple, restriction enzymes ಡಬಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ ಡಿ ಎನ್ ಎ ಮೇಲೆ ಆಕ್ಟ್ ಮಾಡ್ತಕ್ಕಂಥದ್ದು. ಸೊ ಯಾವುದೇ ರ್ಯಾಂಡಮ್ ಪಾರ್ಟ್ ಅಲ್ಲಿ ಎನಿ ಪಾರ್ಟ್ ಅಲ್ಲಿ ಈ ಒಂದು ಡಿ ಎನ್ ಎಸ್ ಡಿ ಎನ್ ಎ ಕಟ್ ಮಾಡೋದಿಲ್ಲ. ಸ್ಪೆಸಿಫಿಕ್ ಸೈಟ್ಸ್ ಇರುತ್ತೆ. ಒಂದು ಗಿವನ್ ಸೀಕ್ವೆನ್ಸ್ ಆಫ್ ಡಿ ಎನ್ ಎ ಕೊಟ್ಟಿದ್ದೀವಿ ಅಂತಂದ್ರೆ ಆ ಡಿ ಎನ್ ಎ ನಲ್ಲಿ ಸ್ಪೆಸಿಫಿಕ್ ಸೀಕ್ವೆನ್ಸ್ ನ ಐಡೆಂಟಿಫೈ ಮಾಡಿ ಆ ಸ್ಪೆಸಿಫಿಕ್ ಪಾಯಿಂಟ್ ಅಲ್ಲಿ ಕಟ್ ಮಾಡ್ತಕ್ಕಂಥ ಒಂದು ಸ್ಪೆಸಿಫಿಟಿ ಇದೆ ಅಥವಾ ರೆಸ್ಟ್ರಿಕ್ಟೆಡ್ ಸೈಟ್ ಇದೆ. ಹಾಗಾಗಿ ಆ ಎನ್ಸೈಮ್ಸ್ ನ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎನ್ಸೈಮ್ಸ್ ಅಂತ ಕರಿತಾ ಇದೀವಿ. The enzymes are referred restriction enzymes because these act on double-stranded DNA at a specific site in the specific sequence of DNA. Hence, the enzymes are referred as restriction enzymes. Right. So, what are these recognition sequences and restriction site? ಏನಾಗಾದ್ರೆ fine. Before that, restriction enzymes further they are into two types. So first important point in note ಮಾಡಬೇಕಾಗಿರೋದು ಅಂತಂದ್ರೆ restriction enzymes act on double stranded DNA, most important point, and they act at a specific site in the specific sequence of DNA. ಸೊ ಮೂರು ಪಾಯಿಂಟ್ ನ ನಾವು ಇಲ್ಲಿ ನೋಟ್ ಮಾಡ್ಕೋಬಹುದು. ಫಸ್ಟ್ ಪಾಯಿಂಟ್ ಏನು ಅಂತಂದ್ರೆ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎನ್ಸೈಮ್ಸ್ ಡಬಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ ಡಿ ಎನ್ ಎ ಮೇಲೆ ಆಕ್ಟ್ ಮಾಡ್ತಕ್ಕಂಥ ಎನ್ಸೈಮ್ಸ್. ಸೆಕೆಂಡ್ ಪಾಯಿಂಟ್ ಏನು, ಸ್ಪೆಸಿಫಿಕ್ ಸೀಕ್ವೆನ್ಸ್ ನ ಐಡೆಂಟಿಫೈ ಮಾಡ್ತಕ್ಕಂಥದ್ದು. ಡಿ ಎನ್ ಎ ನಲ್ಲಿ ರ್ಯಾಂಡಮ್ ಸೀಕ್ವೆನ್ಸ್ ಅಲ್ಲಿ ಕಟ್ ಮಾಡೋದಿಲ್ಲ. ಪ್ರತಿ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎನ್ಸೈಮ್ಸ್ಗೂ ಅದರದೇ ಆದಂತ ಒಂದು ಸ್ಪೆಸಿಫಿಕ್ ಸೀಕ್ವೆನ್ಸ್ ಇರುತ್ತೆ, ಐಡೆಂಟಿಫೈಯಿಂಗ್ ಸೀಕ್ವೆನ್ಸ್ ಇರುತ್ತೆ. ಆ ಸ್ಪೆಸಿಫಿಕ್ ಸೀಕ್ವೆನ್ಸ್ ನ ಐಡೆಂಟಿಫೈ ಮಾಡುತ್ತೆ. ಆ ಸ್ಪೆಸಿಫಿಕ್ ಸೀಕ್ವೆನ್ಸ್ ಅಲ್ಲೂ ಕೂಡ ಸ್ಪೆಸಿಫಿಕ್ ಪಾಯಿಂಟ್ ಅಲ್ಲಿ ಕಟ್ ಮಾಡ್ತಕ್ಕಂಥ ಎಬಿಲಿಟಿ ಇರುತ್ತೆ. ಸೊ ಈ ಮೂರು ಪಾಯಿಂಟ್ ನ ನಾವು ನೆನ್ಪಿಟ್ಕೋಬೇಕು. ಸೊ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎನ್ಸೈಮ್ಸ್ ಫರ್ದರ್ ದೇರ್ ಇಂಟು ಟೂ ಟೈಪ್ಸ್. What are they? Restriction exonucleases and restriction endonuclease. So restriction exonuclease are the enzymes, restriction enzymes, which act at terminals. As the name itself indicates, exo refers to terminals. In a given sequence of DNA, the enzyme will be identifying a specific sequence. In that specific sequence, recognition sequence, they cut the DNA at the terminals. Hence, the enzymes are referred as restriction exonucleases. So, whereas the restriction endonucleases are the one which recognize specific sequence and they cut the DNA at the middle part of the recognition sequence. A recognition sequence is the one which is identified by the restriction enzyme, and the enzyme identifies this particular sequence and they cleave this particular DNA at a middle part or the center part of the recognition sequence. Hence, the enzymes are referred as restriction endonucleases. The restriction endonucleases are preferred over the restriction exonucleases for the reason that the restriction endonucleases they produce the sticky end or staggered end fragments. For example, you see, uh, if there is a restriction digestion, after the cleavage, the resulting fragments would be having the overhanging fragments or the sticky ended fragments. So the fragments resulting after the restriction digestion will have the sticky ends or the staggered ends. So this sticky ends or the staggered ends enables the firm ligation of the two segments of DNA. Hence, the restriction endonucleases are preferred over the restriction exonucleases. So whatever the enzymes we are using for cutting and isolating the desired DNA in our DNA technology, they are restriction endonucleases. Right. Okay. Uh, Restriction endonuclease are the one which are actually preferred over restriction exonuclease. The enzymes which are used in RDNA technology are the restriction endonucleases. Right. In general, I told you that restriction enzymes identify specific sequence of the DNA, and that specific sequence is referred as a recognition sequence. Sequence of that's a specific sequence of nucleotides of DNA identified by restriction endonuclease. So the sequence of nucleotides present in the DNA which are identified by the restriction endonuclease, and that particular sequence is referred as recognition sequence. So there is specific feature or certain unique property of this recognition sequence. ಸೊ ಈ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎನ್ಸೈಮ್ಸ್ ಸ್ಪೆಸಿಫಿಕ್ ಸೀಕ್ವೆನ್ಸ್ ನ ಐಡೆಂಟಿಫೈ ಮಾಡುತ್ತೆ ಒಂದು ಗಿವನ್ DNA ನಲ್ಲಿ ಅಂತಂದಾಗ, ಆ ಐಡೆಂಟಿಫೈ ಮಾಡ್ತಕ್ಕಂತ ಸ್ಪೆಸಿಫಿಕ್ ಸೀಕ್ವೆನ್ಸ್ ಒಂದು ಯುನಿಕ್ ಪ್ರಾಪರ್ಟಿ ಇರುತ್ತೆ, ಒಂದು ಸ್ಪೆಷಲ್ ಕ್ಯಾರೆಕ್ಟರಿಸ್ಟಿಕ್ ಇರುತ್ತೆ. ವಾಟ್ ಇಸ್ ದಟ್? The recognition sequences are generally palindromic in nature. The recognition sequences are generally palindromic in nature. What are palindromic sequences then? So palindromic sequence is one wherein it is the inverted repeats of nitrogen bases in the double stranded DNA, inverted repeats of nitrogen bases on the two strands of given DNA. Inverted repeats of nitrogen bases on the two strands of given DNA. Otherwise, the sequence of nucleotides or the nitrogen bases on one strand is similar to the sequence of nitrogen bases on the other strand when they are read in the opposite direction. When they are read in the opposite direction, I'm saying direction, say from left to right or right to left. If you are reading the nitrogen base of the double stranded DNA, then the nitrogen base is on one strand from left to right. From left to right, the other strand you should read it from right to left. For example, you see, so this is one particular sequence. Nitrogen bases are like GAATTC. One strand ಅಲ್ಲಿ, that is 5 prime 3 prime strand ಅಲ್ಲಿ ಲೆಫ್ಟ್ ಇಂದ ರೈಟ್ ಗೆ ನೀವು ರೀಡ್ ಮಾಡುವಾಗ GAATTC. ಇನ್ನೊಂದು ಸ್ಟ್ಯಾಂಡ್ ಅಲ್ಲಿ ರೈಟ್ ಇಂದ ಲೆಫ್ಟ್ ಗೆ ರೀಡ್ ಮಾಡುವಾಗ GAATTC ಸೇಮ್ ಇದೆ. ಸೊ ಕಾಂಪ್ಲಿಮೆಂಟರಿಟಿ ಇದೆ. Guanine pairs with cytosine, adenine, thymine, adenine, thymine. Similarly, thymidine, adenine, thymidine, adenine, cytosine, and guanine. There is a complementarity. But when you are reading from left to right on one strand and right to left on the other strand, the sequence remains same. Otherwise, you can say the sequence of nitrogen bases on one strand is similar to on the other strand when they are read in same orientation. It is important. ಅಪೋಸಿಟ್ ಡೈರೆಕ್ಷನ್ ಹೇಳ್ತೀವಿ, ಡೈರೆಕ್ಷನ್ ಅಂತಂದ್ರೆ ಲೆಫ್ಟ್ ಟು ರೈಟ್, ರೈಟ್ ಟು ಲೆಫ್ಟ್ ಅಂತ ಅರ್ಥ ಆಯ್ತು. ಓರಿಯಂಟೇಶನ್ ಮೀನ್ಸ್ 5 prime to 3 prime, 3 prime to 5 prime ಅಂತ ಹೇಳ್ತೀವಿ. If you are reading according to orientation, it should be in same orientation. ನೋಡಿ, ಒಂದು ಸ್ಟ್ಯಾಂಡ್ ನ ನೀವು 5 prime 3 prime ಅಲ್ಲಿ ಓದ್ತಾ ಇದ್ದೀರಾ, GAATTC ಅಂತ ಸೀಕ್ವೆನ್ಸ್ ಇದೆ ಅಂತಂದ್ರೆ, ಇನ್ನೊಂದು ಸ್ಟ್ಯಾಂಡ್ ಅಲ್ಲಿ ನೀವು ಅದನ್ನ ಸೇಮ್ ಓರಿಯಂಟೇಶನ್ ಅಲ್ಲಿ ರೀಡ್ ಮಾಡಬೇಕಾಗುತ್ತೆ, that is 5 prime to 3 prime. So palindromic sequences are the inverted repeats of nitrogen bases in the double stranded DNA. Very simple. Otherwise, the nitrogen bases of one strand is similar to the other strand when they are read in opposite direction. Otherwise, when they are read in same orientation. So this is one example of palindromic sequence which is identified by the enzyme called EcoR1 restriction enzyme. So in order to understand this, what exactly is palindromic, there are certain palindromic words which are actually mentioned in the textbook, such as Malayalam. So this is a palindromic word, what we can say. So left to right if you read, and from right to left you read the same word or the sound, what we can get it. So similarly, madam, Gadag, Liril. So all these are the examples of palindromic words. But palindromic sequence ಅಂತಂದಾಗ ಡಬಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ ಸೀಕ್ವೆನ್ಸ್ ಇರುತ್ತೆ. ಇಲ್ಲಿ ಒಂದೇ ಸೀಕ್ವೆನ್ಸ್ ನ ಅಪೋಸಿಟ್ ವರ್ಡ್ ತರ ನಾವು ಇಲ್ಲಿ ರೀಡ್ ಮಾಡ್ತಾ ಇಲ್ಲ, ಕನ್ಫ್ಯೂಸ್ ಆಗಬಾರ್ದು. ಅಲ್ಲಿ ಮಲಯಾಳಂ ಅಂತ ತಗೊಂಡು, ಮಲಯಾಳಂ ಅಂದ್ರೆ ಆ ವರ್ಡ್ ನ ಲೆಫ್ಟ್ ಟು ಲೆಫ್ಟ್ ಓದಿದ್ವಿ ನಾವು. ಬಟ್ ಇಲ್ಲಿ ಪ್ಯಾಲಿಂಡ್ರೋಮಿಕ್ ಸೀಕ್ವೆನ್ಸ್ ಅಂತಂದಾಗ ಡಬಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ DNA ಫಸ್ಟ್ ಇಂಪಾರ್ಟೆಂಟ್ ಪಾಯಿಂಟ್. ಸೊ ಒಂದು ಸ್ಟ್ಯಾಂಡ್ ಅಲ್ಲಿ ಇರ್ತಕ್ಕಂಥ ಸೀಕ್ವೆನ್ಸ್ ಒಂದು ಡೈರೆಕ್ಷನ್ ಅಲ್ಲಿ ಏನು ಒಂದು ಸೀಕ್ವೆನ್ಸ್ ಇರುತ್ತೆ, ಅದೇ ಸೀಕ್ವೆನ್ಸ್ ಇನ್ನೊಂದು ಸ್ಟ್ಯಾಂಡ್ ಅಲ್ಲಿ ಅಪೋಸಿಟ್ ಡೈರೆಕ್ಷನ್ ಅಲ್ಲಿ ಓದಿದಾಗ ನಮಗೆ ಸಿಗುತ್ತೆ. GAATTC ಐದರ್ ಅಪೋಸಿಟ್ ಡೈರೆಕ್ಷನ್ ಆರ್ ಸೇಮ್ ಓರಿಯಂಟೇಶನ್. Right. Then what is a restriction site then? So restriction site is the specific site within the recognition sequence where the enzyme induces cut, where it the cleaves the phosphodiester bond. So this is the recognition sequence, entire sequence GAA-TTC, GAA-TTC. Within the sequence, the enzymes identify specific sequence. This is an example. Okay. This is an example of palindromic sequence or the recognition sequence for EcoR1. So there are different types of restriction enzymes. Each restriction enzyme will have its own recognition sequence, its own restriction site. So one the example no GAA-TTC ಅನ್ನೋದು palindromic sequence ಅಥವಾ recognition sequence. ಯಾವ enzyme ಗೆ EcoR1 ಅನ್ನೋ restriction enzyme. So within this recognition sequence, the enzyme induces cut between specific sites, between specific nucleotides. So for example, nodi between G and A cut ಮಾಡ್ತಾ ಇದೆ EcoR1. So this specific site is referred as restriction site. Right. So today there are nearly more than 900 restriction endonucleases as isolated from nearly 230 different strains of bacteria. So point ನೆನ್ಪಿಡ್ಬೇಕಾಗಿರೋದೇನು ನಿಯರ್ಲಿ 900 ಡಿಫರೆಂಟ್ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎನ್ಸೈಮ್ಸ್ ಐಸೋಲೇಟ್ ಮಾಡಿರ್ತಕ್ಕಂಥದ್ದು ಅಂಡ್ 230 ಡಿಫರೆಂಟ್ ಸ್ಟ್ರೈನ್ಸ್ ಆಫ್ ಬ್ಯಾಕ್ಟೀರಿಯಿಂದ ಐಸೋಲೇಟ್ ಮಾಡಿರ್ತಕ್ಕಂಥದ್ದು. ಪ್ರತಿ ಒಂದು ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎನ್ಸೈಮ್ಸ್ಗೂ ಅದರದ್ದೇ ಆದಂತ ರೆಕಗ್ನಿಷನ್ ಸೀಕ್ವೆನ್ಸ್ ಇರುತ್ತೆ, ಅದರದ್ದೇ ಆದಂತ ಸ್ಪೆಸಿಫಿಕ್ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಸೈಟ್ ಇರುತ್ತೆ. There is no restriction enzyme. There is no restriction enzyme. So wherein they can identify same recognition sequence, though they identify, they won't cleave the same restriction site. ಯಾವುದೇ ಎರಡು ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎನ್ಸೈಮ್ಸ್ ಸೇಮ್ ರೆಕಗ್ನಿಷನ್ ಸೀಕ್ವೆನ್ಸ್ ಐಡೆಂಟಿಫೈ ಮಾಡ್ತಕ್ಕಂಥದ್ದು ಇರೋದಿಲ್ಲ. ರೆಕಗ್ನಿಷನ್ ಸೀಕ್ವೆನ್ಸ್ ಸೇಮ್ ಇದ್ರೂ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಸೈಟ್ ಅಂತೂ ಡಿಫ್ರೆಂಟ್ ಇರಲೇಬೇಕಾಗುತ್ತೆ. So there are no two enzymes wherein they can cleave between same restriction site. So nearly 900 different restriction endonuclease has been isolated and from 230 different strains of bacteria. So when was this actually discovered? What was the discovery of restriction enzymes or the endonucleases? So scientists by name, three different scientists actually, Smith, Arbor and Nathan. Smith, Arbor and Nathan in the year 1963, so they identified, they discovered these particular restriction enzymes. So in E. coli cells, so there were two types of enzymes which are isolated as a defensive tools. The first class of enzymes were called methylases and the second were called restriction enzymes. So bacteria protected itself from the bacteriophages, the bacteriophages. So wherein they protected their own self DNA by methylating themselves. So methylases are the enzymes which methylate the native DNA in order to protect themselves from uh, the fast DNA, and the restriction enzymes are the one which are present in the bacteria. So where they act as a defensive tool against the first DNA. So these were the two class of enzymes which were identified. Among this, one of the type of enzymes were the restriction enzymes, so which were identified, which were discovered by Smith, Arbor and Nathan in the year 1963. And the first restriction endonuclease discovered was HIN2. This is also important. So which is the first, which of the following is the first discovered restriction endonuclease? So you should be knowing HIN2, Haemophilus influenza D strain 2, right? So let us see what is the nomenclature of restriction enzymes. So there are nearly 900 different restriction enzymes. So there must be some nomenclature or procedure for naming these restriction enzymes. So the first point is the restriction enzymes are named based on bacteria from which they are isolated. Basically, the restriction enzymes are present in the bacteria. Different restriction enzymes are present in different types of bacteria. So the first point what we need to consider is from which bacteria the enzyme has been isolated. ಬೇರೆ ಬೇರೆ ಬ್ಯಾಕ್ಟೀರಿಯಾನಲ್ಲಿ ಬೇರೆ ಬೇರೆ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎನ್ಸೈಮ್ಸ್ ಇರುತ್ತೆ. ಸೊ ನೇಮಿಂಗ್ ಕೊಡುವಾಗ ಬೇಸಿಕಲಿ ಯಾವ ಒಂದು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಇಂದ ಒಂದು ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎನ್ಸೈಮ್ ಐಸೋಲೇಟ್ ಮಾಡ್ತಾ ಇದೀವಿ ಆ ಬ್ಯಾಕ್ಟೀರಿಯಾ ನ ಕನ್ಸಿಡರ್ ಮಾಡ್ತಾ ಇದೀವಿ. So based on bacteria from which they are isolated. Okay. The name of enzyme will be given like this. First letter of the enzyme is from first letter of enzyme is from first letter of the genus name. So we know that every living organism is given a binomial nomenclature. The first name will be the genus and the second name will be the species. So a bacteria will also be having a binomial nomenclature. So wherein it will be having a genus and species name. The first letter of the enzyme. So while naming the enzyme, the first letter of the enzyme will be from the first letter of the genus name of that bacteria. I'll give an example for that. At the end of this particular explanation. Okay. So first letter of the enzyme is from first letter of the genus. Okay. Next two letters of the enzyme is from first two letters of species. The first we are considering the first letter of the genus, and next two letters in the enzyme will be from the first two letters of the species name. Okay. Okay. Next, followed by the enzyme name will be followed by strain name and finally Roman number represents order of isolation. So Roman number represent the order of isolation of the enzyme. For example, you see EcoR1. This is an restriction enzyme. The name of the restriction enzyme. Here, E refers to the first letter of the enzyme, that is E, is from the first letter of the genus from the bacteria from which it is isolated, that is Escherichia. And the next two letters, CO, are from the first two letters of the species of the bacteria from which they are isolated, that is coli. And R refers to strain, RY. And Roman number 1 refers to the order in which it is isolated, that means 1 refers to first isolated. From Escherichia coli bacteria, the restriction enzyme was isolated. The first restriction enzyme was isolated and the name was given from the first letter of the genus, that is Escherichia, E was considered, and the next the first two letters of the species was considered, and the strain name is considered, and Roman number 1 is given in order to depict the order of isolation. So eco R1 is an example in order to explain this particular nomenclature. So E refers to Escherichia, CO refers to coli, R refers to the strain, and the Roman number 1 refers to the order of isolation. Right. So there are different types of enzymes such as EcoR1, EcoR2, EcoR3, etc. Similarly, HIND1, HIND2, HIND3, etc. So I just mentioned HIND2 was the first discovered restriction enzyme, HIND2. Then we represent EcoR1 as first isolated, HIND1, HIND2, HIND3. So you'll confuse ಆಗ್ಬೋದು ನಮಗೆ. HIND1, HIND2, HIND3 ಅಂತ ಇದೆ. ರೋಮನ್ ನಂಬರ್ ಅನ್ನೋದು ಆರ್ಡರ್ ಆಫ್ ಐಸೋಲೇಷನ್ ನ ರೆಪ್ರೆಸೆಂಟ್ ಮಾಡುತ್ತೆ. ಮತ್ತೆ ಫಸ್ಟ್ ಡಿಸ್ಕವರ್ಡ್ ಅನ್ನೋದನ್ನ ನಾವು HIND2 ಅಂತ ಇದೀವಲ್ಲ, ಹೇಗೆ ಇದು? Of course, first discovered was HIND2. Roman number doesn't mean the discovery. HIND1, HIND2, HIND3. So this Roman number 1, 2, 3, this represent order of isolation. ಅಂದ್ರೆ ಹಿಮೋಫಿಲಸ್ ಇನ್ಫ್ಲೂಯೆನ್ಸಾ ಡಿ ಸ್ಟ್ರೇನ್ ಅನ್ನ ಒಂದು ಬ್ಯಾಕ್ಟೀರಿಯಿಂದ ಫಸ್ಟ್ ಒಂದು ಎಂಜೈಮ್ ಐಸೋಲೇಟ್ ಮಾಡಿದ್ರು. ಅದಾದ್ಮೇಲೆ ಇನ್ನೊಂದು ಯಾವ್ದೋ ಐಸೋಲೇಟ್ ಮಾಡಿದ್ರು, ಅದನ್ನ 2 ಅಂತ ಹೇಳಿದ್ರು. ಇನ್ನೊಂದು ಯಾವ್ದೋ ಥರ್ಡ್ ಎಂಜೈಮ್ ಐಸೋಲೇಟ್ ಮಾಡಿದ್ರು, ಅದನ್ನ 3, ರೋಮನ್ ನಂಬರ್ 3 ಅಂತ ರೆಪ್ರೆಸೆಂಟ್ ಮಾಡಿದ್ರು. ಸೊ ಅಮಾಂಗ್ ಆಲ್ ದೀಸ್ 3, ದಿ ಫಸ್ಟ್ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಜೈಮ್ ಆಕ್ಟಿವಿಟಿ, the first restriction endonuclease activity was identified for this enzyme. ಈ ಮೂರನ್ನ ಐಸೋಲೇಟ್ ಮಾಡಿದ್ರು. ಇದು ಫಸ್ಟ್ ಐಸೋಲೇಟೆಡ್, ಸೆಕೆಂಡ್ ಐಸೋಲೇಟೆಡ್, ಥರ್ಡ್ ಐಸೋಲೇಟೆಡ್. ಈ ಮೂರರಲ್ಲಿ ಆ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸ್ ಎಬಿಲಿಟಿ ಅಥವಾ ಆಕ್ಟಿವಿಟಿ ಇದೆ ಅಂತ ಐಡೆಂಟಿಫೈ ಮಾಡಿದ್ದು, ಡಿಸ್ಕವರ್ ಮಾಡಿದ್ದು HIND2ಗೆ. ಹಾಗಾಗಿ ಫಸ್ಟ್ ಡಿಸ್ಕವರ್ಡ್ HIND2 ಅಂತ ಹೇಳ್ತಾರೆ. Well, say the recognition sequence for EcoR1. I just so this is also very important point to be remembered for the competitive exams. So they will ask you which of the following is the recognition sequence for EcoR1. So remember, GAATTC and the orientation is 5 prime 3 prime. If you are writing the orientation as 5 prime 3 prime, it should be GAATTC. Clear? So the other sequence will be CTTAAG, that is in 3 prime 5 prime. So GAATTC in 5 prime 3 prime. Remember, GA, they may give option like GAATTC with orientation such as 3 prime 5 prime. Okay, so this is very careful, you should be remembering. The sequence is GAATTC and the orientation is 5 prime 3 prime. So this is most important point what you need to remember for the MCQs. Okay, that's about the restriction enzymes or the endonuclease. So the restriction endonuclease, they are commonly called as molecular seizures. So without the restriction endonucleases, so we cannot imagine, there is no possibility of the existence of rDNA technology. ಸೊ ಈ ಮಾಲಿಕ್ಯುಲರ್ ಸೀಸರ್ಸ್ ಅಥವಾ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಡೋನ್ಯೂಕ್ಲಿಯೇಸಸ್ ಇಲ್ಲ ಅಂತಂದ್ರೆ ಈ RDNA ಟೆಕ್ನಾಲಜಿನೇ ಇರೋದಿಲ್ಲ. ಸೊ ಬೇಸಿಕಲಿ ಮಾಲಿಕ್ಯುಲರ್ ಟೈಲರಿಂಗ್ ಅಂತ ಹೇಳಿದೆ. ಸೊ ಯಾವುದೇ ಟೈಲರ್ ಅಂದ ತಕ್ಷಣ ದಿ ಬೇಸಿಕ್ ಟೂಲ್ ಸೀಸರ್ ಇರಬೇಕಾಗುತ್ತೆ. ಸೊ ಇಲ್ಲಿ ಕೂಡ ಮಾಲಿಕ್ಯುಲರ್ ಟೈಲರಿಂಗ್ ಅಂತ ಕರಿತಾ ಇದೀವಿ ಅಂತ ಹೇಳಿದಾಗ ಫಸ್ಟ್ DNA ಕಟ್ ಮಾಡ್ಕೊಂಡ್ರೆ ನಾವು ಡಿಸೈರ್ಡ್ ಜೀನ್ ಐಸೋಲೇಟ್ ಮಾಡಿದ್ರೆ ನಾವು ಮುಂದಿನ ಪ್ರೊಸೀಜರ್ ಮಾಡಬೇಕಾಗುತ್ತೆ. ಸೊ ಸೊ restriction enzymes are mandatory for our DNA technology. Okay, next enzymes are the ligases. So ligases are called molecular glues. So these are the enzymes which are used to ligate two segments of DNA. The enzymes are used to ligate two segments of DNA. So these create the phosphodiester bonds between the segments of DNA. So the most commonly used the DNA ligases are from T4 phase, hence they are called T4 ligase. It's actually not mentioned in the textbook, just for information. So ligases are the molecular glues, the enzymes which are used to join the two segments or ligate the two segments of the DNA, and they produce the phosphodiester bond between the two segments of the DNA. And the most commonly used ligase enzymes in our DNA technology are isolated from T4 phase, and they are referred as T4 ligases. Okay, then the polymerase enzymes. So most commonly important, the enzymes DNA polymerases will be used. DNA polymerase enzymes are used in our DNA technology, and the other class of enzymes are DNA modifying enzymes. So this group of enzymes, DNA modifying enzymes, they're not mentioned in the textbook. There is no information regarding this, but however, just mentioning what are these DNA modifying enzymes. So during the process of construction of recombinant DNA, so various enzymes are used for certain processes such as removing phosphate from the 5 prime and addition of phosphate or digestion of single strand of DNA. So all those enzymes are referred as DNA modifying enzymes such as alkaline phosphatases, alkaline phosphatase enzyme, phosphokinase enzymes, then S1 nuclease. So alkaline phosphatases are the enzymes which remove phosphate from 5 prime. Phosphokinases are the one which add the phosphate to the 5 prime end, and S1 nucleases are the one which actually digest the single strand DNA. This is actually not mentioned in the textbook. Just on the information ಗೊತ್ತಿರಲಿ note ಮಾಡ್ಕೊಂಡಿರಿ ಅಷ್ಟೇ. Okay. So next important tools for RDNA technology are the vectors. So vectors, these are commonly called as molecular vehicles. So we have isolated the desired gene. When you have the desired gene, now you need to transfer this desired gene into host cell. So in order to transfer, so immediately when I say transfer, there must be some mechanism of transportation. So in order to transfer this desired gene into the host cell, there must be some structures, some medium for transportation or transferring. So one of such mechanism or the transportation or transfer of gene can be achieved by using the vectors. So vectors are basically, these are the DNA molecules used to transfer desired gene into host cell. So the DNA molecules which are used to transfer the desired gene into the host cell, and those DNA molecules are referred as vectors. Okay. So not all DNA molecule or any DNA molecule can be used as vectors. There must be certain properties. So very simple, we use animals for transportation. So animals ಅಲ್ಲಿ ಯಾವುದು such as the ox, or it may be horse, or it may be camel, or it may be yak, or it may be even elephants in case of carrying the logs of wood in forest. ಸೊ ಈ ಅನಿಮಲ್ಸ್ ನಾವು ಟ್ರಾನ್ಸ್ಪೋರ್ಟೇಷನ್ ಯೂಸ್ ಮಾಡ್ತಾ ಇದೀವಿ. ಎಲ್ಲ ಅನಿಮಲ್ಸ್ ನ ಟ್ರಾನ್ಸ್ಪೋರ್ಟೇಷನ್ ಯೂಸ್ ಮಾಡ್ತಾ ಇಲ್ಲ. ಸೊ ಈ ಕೆಲವೊಂದು ಪ್ರಾಪರ್ಟೀಸ್ ಇರಬೇಕಾಗುತ್ತೆ ಆ ಒಂದು ಟ್ರಾನ್ಸ್ಪೋರ್ಟೇಷನ್ಗೆ ನಾವು ಅನಿಮಲ್ ನ ಯೂಸ್ ಮಾಡ್ತಾ ಇದ್ದೀವಿ ಅಂತ. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಡೆಸರ್ಟ್ ಅಲ್ಲಿ ಯಾವ ಒಂದು ಅನಿಮಲ್ ಕಂಫರ್ಟ್ ಆಗಿ ವಾಕ್ ಮಾಡಬಹುದು ಅಂತ ಒಂದು ಅನಿಮಲ್ ನ ನಾವು ಯೂಸ್ ಮಾಡ್ತೀವಿ. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಯಾವುದು, ನಾವು ಕ್ಯಾಮಲ್ ನ ಡೆಸರ್ಟ್ ಅಲ್ಲಿ ಟ್ರಾನ್ಸ್ಪೋರ್ಟೇಷನ್ ಯೂಸ್ ಮಾಡ್ತೇವೆ. So similarly, DNA molecule which is used as a vector should have certain properties. Not all DNA molecules can be used. So the vectors, the DNA molecule which we use can be a plasmid or it may be a fast DNA. So we may either use this plasmid DNA or phase DNA as vector. But if at all we are using plasmid or phase DNA, they have to satisfy certain properties. So what are those? Certain salient features. ಕೆಲವೊಂದು ಪ್ರಾಪರ್ಟೀಸ್ ಇರಬೇಕಾಗುತ್ತೆ ವೆಕ್ಟರ್ ಆಗಿ ಯೂಸ್ ಮಾಡಬೇಕು ಅಂತ. ಸೊ ಏನೇನದು? ಇಟ್ ಶುಡ್ ಬಿ ಈಸಿ ಟು ಐಸೋಲೇಟ್ ಅಂಡ್ ಟ್ರಾನ್ಸ್ಫರ್. Should be easy to isolate and transfer. Should consist origin of replication. So a vector must have origin of replication in order to ensure the replication of the gene which is carrying. Gene which it carries. So ಯಾವುದೇ ಒಂದು ವೆಕ್ಟರ್ ಒಂದು ಜೀನ್ ನ ತಗೊಂಡು ಹೋಗ್ತಾ ಇದೆ ಅಂದ್ರೆ ಆ ಜೀನ್ ನ ಆ ಒಂದು ಹೋಸ್ಟ್ ಸೆಲ್ ಅಲ್ಲಿ ರೆಪ್ಲಿಕೇಶನ್ ನ ಒಂದು ಫೆಸಿಲಿಟೇಟ್ ಮಾಡ್ತಕ್ಕಂತ ಒಂದು ಕ್ಯಾರೆಕ್ಟರ್ ಇರಬೇಕು. ಸೊ ರೆಪ್ಲಿಕೇಶನ್ ಫೆಸಿಲಿಟೇಟ್ ಸಪೋರ್ಟ್ ಮಾಡಬೇಕು ಅಂತಂದಾಗ ವೆಕ್ಟರ್ ಅಲ್ಲಿ ಆರಿಜಿನ್ ಆಫ್ ರೆಪ್ಲಿಕೇಶನ್ ಇರಬೇಕಾಗುತ್ತೆ. ಓಕೆ, it should consist at least 2 marker genes. See, marker genes are the one which are used to identify the transfer of desired gene. ಯಾವ ಸೆಲ್ ಅಲ್ಲಿ ನಮ್ಮ ಡಿಸೈರ್ಡ್ ಜೀನ್ ಎಂಟ್ರಿ ಆಗಿದೆ, ಯಾವ್ದ್ರಲ್ಲಿ ಆಗಿಲ್ಲ ಅನ್ನೋದನ್ನ ಐಡೆಂಟಿಫೈ ಮಾಡಬೇಕು ಅಂತಂದಾಗ ಒಂದು ಮೆಕ್ಯಾನಿಸಮ್ ಬೇಕಾಗುತ್ತೆ. ಕಣ್ಣಿಗೆ ಕಾಣಿಸೋದಿಲ್ಲ, ಸೆಲ್ ಅಂತಂದ್ರೂ ಕಣ್ಣಿಗೆ ಕಾಣಿಸಲ್ಲ, ಜೀನ್ಸ್ ಅಂದ್ರೂ ಕಾಣಿಸ್ತಾ ಇಲ್ಲ. ಸೊ ಯಾವ ಸೆಲ್ ಅಲ್ಲಿ ಡಿಸೈರ್ಡ್ ಜೀನ್ ಎಂಟ್ರಿ ಆಗಿರುತ್ತೆ, ಯಾವ್ದ್ರಲ್ಲಿ ಆಗಿರೋದಿಲ್ಲ ಅನ್ನೋದನ್ನ ಐಡೆಂಟಿಫೈ ಮಾಡೋದಕ್ಕೆ ಒಂದು ಮೆಕ್ಯಾನಿಸಮ್ ಬೇಕಾಗಿರುತ್ತೆ. ಅದನ್ನೇ ನಾವು ಒಂದು ಮಾರ್ಕರ್ ಜೀನ್ಸ್ ಇಂದ ಅಚೀವ್ ಮಾಡ್ತೀವಿ. ಸೊ ದ ಜೀನ್ಸ್ ವಿಚ್ ಆರ್ ಯೂಸ್ ಟು ಐಡೆಂಟಿಫೈ ಟ್ರಾನ್ಸ್ಫಾರ್ಮ್ಡ್ ಸೆಲ್ಸ್ or to differentiate transformed and non-transformed are referred as marker genes. We have 2 types of marker genes there, selectable and scorable marker genes. The details we'll be discussing in the process. Okay, then so it should have multiple cloning sites. A vector should have multiple cloning sites. That means it should have a point at which it can be cleaved and the desired gene can be ligated. So there must be multiple cloning site. That means a vector should have unique restriction site for as many restriction enzymes as possible. Restriction endonucleases. The vector should have unique restriction site for as many restriction enzymes as possible. So vectors basically they are the DNA molecules which are used to transfer the desired gene into the host cell, and they are used as vehicles to transfer the desired gene, as they are the biomolecules, hence they are referred as molecular vehicles. Generally, plasmids or the phase DNAs are used as the vectors. All DNA or any DNA cannot be used as vector. They must have certain properties. It should be easy to isolate and transfer. It should consist origin of replication in order to support the replication of the gene which it carries into the host cell. It should consist at least two marker genes in order to differentiate transformed and non-transformed cells. It should have multiple cloning sites, unique restriction sites for as many restriction endonuclease as possible. Right. So there are few examples of these vectors, such as the PBR322 and the TI plasmid. What are the different types of vectors which are used to transfer the desired gene into the bacteria, and the gene into the plant cell, and the gene into the animal cell. So ಒಂದೊಂದು ಸೆಲ್ಗೂ ಸ್ಪೆಸಿಫಿಕ್ ಸೆಲ್ಗೂ ಒಂದು ಸ್ಪೆಸಿಫಿಕ್ ಟೈಪ್ ಆಫ್ ವೆಕ್ಟರ್ ಇರುತ್ತೆ. ಸಿಂಪಲ್ ಆಗಿ ಈ ನೀರಿನಲ್ಲಿ ಟ್ರಾನ್ಸ್ಪೋರ್ಟೇಷನ್ ಆಗಬೇಕು ಅಂದಾಗ ಅಲ್ಲಿ ಸ್ಪೆಸಿಫಿಕ್ ವೆಹಿಕಲ್ ಇರುತ್ತೆ, ಲ್ಯಾಂಡ್ ಅಲ್ಲಿ ಅಂದಾಗ ಸ್ಪೆಸಿಫಿಕ್ ವೆಹಿಕಲ್ ಇರುತ್ತೆ, ಅದೇ ತರ ಏರ್ ಅಲ್ಲಿ ಸ್ಪೆಸಿಫಿಕ್ ಟ್ರಾನ್ಸ್ಪೋರ್ಟೇಷನ್ ಮೆಕ್ಯಾನಿಸಮ್ ಇರುತ್ತೆ. ಸೊ ಡಿಪೆಂಡಿಂಗ್ ಆನ್ ವಾಟ್ ಟೈಪ್ ಆಫ್ ದಿ ಹೋಸ್ಟ್ ಸೆಲ್ ವಿ ಆರ್ intended to transform, so based on that there will be specific vectors, so which will be using to transfer the desired gene into the host cell. So those, what are those types of vectors, and what are the some examples, the structure of PBR322, and further concepts will be continuing in the next session. Thank you. Have a nice day.